Control deck, that is Herc away.
Being Herc away.
Control deck, Hercules is past the transom.
Copy.
Control deck, that is tether all out.
Copy, tether all out.
Hey Bob, can you give us a yell when it's okay to turn on the eDNA sampler? All right. But it's not responding.
So what I did just then was set it for this dive.
Control deck, Atalanta is away.
Copy, Atalanta away. Hercules is dive, dive, dive.
Is to look at this window, which is volt of sand plus absorbance. That shows your spectrum, your raw spectrum. And you can just look in this noise region and check it's moving.
Yeah.
Because these will probably be pretty stable for a lot of the dive. You have 4 tabs at the top, and this will show stuff you're interested in, basically.
Roger, I think you can go ahead and do that.
So concentration, this will show trend in concentration and time. It should continuously update. If they turn it off, say there's a ground fault and they want to check something, they turn it off, you just get like a
Zeros
empty
out.
window.
Yeah.
Voltage scan absorbance, this is your actual spectrum. So this is when you'll see this like range of normal spectra,
Yep.
basically. The one difference with this one, this is an image of a methane one. This is an audio slate for dive. Or like a bit. So we're never going to get this real base for hotel 2051. UTC time is 07:49:25. Like way more than should just be Mark. Ocean, and that would be like fluids might be really hot or whatever. So that would be like this was in the Guaymas Basin with methane, and it's like we're not going to see that here. But if for some reason you did, that's like definitely a warning sign. Like in itself, it's not bad, but it's just a bit like surprising. Then the parameter you're really going to want to check to make sure that stage is happy is this one, inlet gas humidity.
Is that going to change color if it gets unhappy?
It doesn't change color.
Okay.
But it will get really high. So usually in a dive, yesterday was at like 20%. That was good. 40% fine, 50, 60. If it gets to like 80%, then the humidity is getting high, and there might be a reason for that. And probably in this region where there's nothing hydrothermal, the most likely thing is like the pump isn't working great.
Okay, stand by 1 sec. Hey, Bob, when you get a second, can you confirm that there's power to the eDNA sampler? Okay. 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 Yes. Yeah, no worries. All right.
If this gets high, it basically means it's unexpectedly warm or the pump isn't working. If it does do that, the easiest thing for you to check first is basically go configuration, open config file editor.
1 second.
Yeah.
Erica, can you log that the eDNA sampler is on and communicating? And after that, that the SAGE sampler is on and working.
I'm a bit hesitant to actually do this because we'll interrupt the data during the nice descent profile.
Don't do it.
But basically you just go configuration, there'll be a green box that says external pump. You click it so it goes red, then you click it so it goes green, then you click OK and it will restart the pump.
Yeah, puffer's talking.
That's going to be the easiest way for you to check. If that doesn't fix it, then you could ask the pilots to do a hard power cycle, and if that doesn't fix it, call me.
Exactly.
But it's not going to get that high on a dive like this. And then for this type of dive, the only other thing you're going to want to be careful of is the other warning sign, by the way, if the pump isn't working, inlet gas pressure will go high because it's not exchanging. But those two things, so that will go to like 1,100. That is its maximum value. That will be going up. Those are the two signs that like the pump's not working. The one thing you need to be, the only possible scenario that would be like a human safety issue is before ascent. These two, this is the pressure inside the housing. It should be really low. 140 millibar is super low. If that, for some random reason, was reading over atmospheric pressure, then the overpressure valve is going to blow on the surface. Like, and it won't blow, it will just be like, psst.
Okay.
It's not going to happen on this dive.
All
That's
right.
like in a hydrothermal dive.
We need 4 bullets with numbers.
That is exactly what you have here. So it's literally like pointing to, it's hard to read because of the red light, but it's literally like pointing to this button. It should always stay pretty constant during live and be at around 100 millibar. That's fine. So humidity should always be less than 80%. And this is generally around 700, 900, whatever it is during a dive.
Yeah, so I might
Yeah.
just kind of like, this one's super important.
Yeah.
This one's super important.
Yeah.
And this one?
This one here is, if that goes high, that's probably the pump's not working.
Okay.
And yeah.
Okay.
That's basically it. I think you're going to have a very uneventful dive in terms of there's nothing hydrothermal, there's nothing hot, there's nothing really gaseous. Don't put it in bubbles. Don't put it in like 50-degree water. You'll be fine. And I'll be up in 3 hours and you can tell me that it was totally fine. This is going to be your most interesting tab. It will show any trends.
Perfect.
CO2.
Let's leave it there.
Okay. And it's saving to a folder. It's doing its own thing. Let me just check that folder is empty. That's your thing. Call me if you need anything.
All right, go to bed.
See you in 3 hours time.
Present.
I will also check in with Annette about, I don't know if she's staying up till my shift.
I'm not sure.
Because I know she was going to talk me through eDNA sampling. So I'll find out about that.
Okay.
Or I go to bed in case she wants me to do that.
Okay. Why is that? Oh. Yeah, gotcha. No, he's gonna cycle power, and then we have to figure out how to turn it back on. Yeah, Val, you see how it stopped, oops. Roger. Okay. Hey, Annette, you listening? All right, we got the Sage back on again. Oh, man. Hey, Johan, what's the depth we're aiming for here? Uh, 1504. Thank you. Perfect. I'll bet money. Did I win? Oh. All right. If we have to run without him, yeah, it's fine. The manip? I mean, yeah, we'll keep going, but I'd love to have it. Yes. Yeah. Maybe I'll put it on. Okay. What's the metal rod coming off to the... Oh, that's the temp rope. Oh, no, I like it. That's fine. Yeah, that is field guides that is on the desktop.
Yeah. Okay, Robert.
Yeah.
Do you mind if I start moving the ship back to our starting point?
You can go for it.
All right, it's about 100 meters at 300. Bridge control.
Check.
Can we move the ship 100 meters at 300, please? Copy that, thank you. Oh, really? Okay.
Alright, so for everyone. Welcome aboard. I know this is our second dive of the day. I'm Stefano Wanga as our communication fellow for today. We're now in our Tutuila Ridge near the Someone Clippers area search area we have on. Welcome to our Emamano Ngatay Fale second expedition for our American Samoa journey. And we're gonna go around the room, introduce yourself, feel comfortable, and see what we can do for our dive today. All right, moving on to my right.
Yeah. Hello. Hey, everybody. It's Adam Sewell. I'm from the University of Rhode Island, the director of the Ocean Exploration Cooperative Institute, which is a collection of uh, research institutions and organizations that focus on helping NOAA achieve its mandate to explore the oceans. And that is definitely what we're doing today. We're going to a spot that no human eyes have ever seen before, which is pretty exciting. This is my, I would say, probably fifth or sixth time out on Nautilus. I'm the watch lead for the 8 to 12 watch, and I'm really excited to have all of you joining along with us. So I will pass it on to Val to introduce herself. No, we can't hear you. Yeah, you gotta tap that one.
Talofa lava. My name is Val Brown. I am the research coordinator at the National Marine Sanctuary of American Samoa. And this is my first cruise aboard EV Nautilus. I'm very excited to be here. And I will pass it over to my right.
Sorry. Hi everyone, my name is Erica Leon Guerrero and I am an ocean science intern on this cruise.
Can you get the mic closer to your mouth, please?
Like that. Yeah, okay.
Like right up on your lips is good.
Cool. Okay, and I'm very excited to be here. I'll pass it to Dave.
Hi, this is Dave. I'm lead video engineer on this cruise and sitting here doing video for the 8 to 12 watch. Hopefully we'll see some interesting stuff when we get down to the bottom. Next to me, I have the video intern, Samantha. Say hi, Samantha.
Hello everyone, my name is Samantha. I'm the video intern for this leg of the Nautilus expedition here in American Samoa. Happy to be here and excited to learn more.
Hi guys, my name is Esteban Juan. I am the ROV intern. I am currently in charge of piloting Atalanta. It is my first cruise on Nautilus. Excited to be here. Ready to see what we can find down in the depths.
And I'm Robert Waters. I'm sitting in the Herc pilot seat. I'm OET's facilities manager and ROV engineer. And I've been on here a few times.
Hey everyone, I'm Johan Becker. I'm the navigator for this 8 to 12 shift. Off the boat, I'm a grad student at the University of Rhode Island in ocean engineering.
Nice. And we are headed to the seafloor at a depth of about 1,500 meters, where we should see the seafloor. And then we're going to move up the ridge towards the island. To me, it looks kind of like a volcanic constructional ridge, but we'll see when we get down there. Kind of sticks up off the seafloor, so it should be a good spot to see the organisms that like to attach themselves to hard substrates, coral sponges and the like, and all the other organisms that associate with them. So pretty exciting. So far on the way down, we're seeing a bunch of little squid and a whole bunch of marine snow and plankton in the water. So it's a pretty productive area and should be great. And we're pretty near, as we come up this ridge, towards a village. Can you tell us what village that is? The one that we
Yes.
yeah?
So we were heading toward a village of Fangasa. Yeah, one of those villages that are located on way back from the downtown area, close to the ridge. And this village, yep, kind of like similar to where I come from on the other eastern part, which is Vatia. And they also have some good historical stories and how they come about and build their community there at that nice spot that we were able to get a closer view.
Yeah, it's a really cool spot. It looks like it has, well, we could only see kind of right into the bay, but it looks like the bay kind of like spreads out back there. Yeah, so really protected bay, and looks like an awesome spot to visit.
one of the most prominent things that this village has been done. It's they're one of the villages with one of the Fautasi, which is one of the big festival that we have here in American Samoa, the Fautasi race, where we have the
Oh,
canoes
yeah.
and rowers. So they have like some multiple times winning the Fautasi race
Whoa.
back then in 2000, and they were also invited to compete in Western Samoa or Inopia
Hmm.
with that same category, Fautasi race, and they also won. So that's one of the most highlights that this
Wow.
village has been proud of,
They're
and
champions.
as of now they're continuing with this sports. So they have been known for being racing on the Fautasi race throughout the races that we have.
And do those races happen often, or like is it kind of a once a year kind of thing?
Usually it's once a year during the Flag Days on April,
Uh-huh.
I think April 14, during that week where we have all the festivities of the celebration of the Flag Days.
Hmm.
And then as of now, we also have other celebrations for each departments.
That's a fish.
So they for those celebration, they decided to have like a Fautasi race because it's been one of the most best sports that we have here. Like,
Yeah.
people get to go out and watch. It's like an off day from work
Uh-huh.
because everybody wants to see the boats
Hmm.
see the boats roaring. So yes, it has been more than once a year
Cool.
race. Yeah.
So also on the vehicle for this dive, we have a couple interesting sensors and samplers that have been integrated with the vehicle and came from the different research groups that are participating in the expedition. We have a sensor called SAGE, which stands for, darn it, I thought it was going to be written down. Sensor for aqueous gases in the environment. So this sensor we have on today measures dissolved carbon dioxide in the water, and we have another one that we'll put on that measures dissolved methane. And it's a pretty cool technology. During the next watch, the person responsible for these sensors will be on watch and can describe the technology in more detail. And then we also have a sampler called the Multi-Puffer. And Annette, if you want to take a moment and describe what that is, that'd be cool.
Hi, this is Annette here. Can you all hear me?
Oh yeah, you've got to introduce yourself because we kind of skipped over you off to the side there.
Sure. I'm Annette Govindarajan. I'm a biologist at the Woods Hole Oceanographic Institution, and I'm interested in the biodiversity of animals in the water column in the deep ocean, and the way that we're looking for these is with environmental DNA, or eDNA as we call it, the forensics of the ocean, where we look for the genetic fingerprints that the animals leave behind as they move through the water. So we've developed a new eDNA sampler that collects these genetic traces by filtering water and does the filtration in situ, so we can mount the samplers on different platforms. We're testing out a new sampler today from Aquatic Labs that we're putting on Hercules, and so this is a really exciting deployment. So hopefully it'll be the first time we can collect samples using the multi-puffer on Hercules.
Thanks, Annette. And those who are watching online, just letting you know you can write in with questions, and if we have the time, we'll try and answer them. So we've now approaching 700 meters, so about halfway to the depth we're trying to get to tonight. Yeah, I was just reminded that while we're diving the ROV this evening, we also have another robot in the water. That's the autonomous underwater vehicle Sentry. It is running a survey just to the west of where we're diving, and it's looking in an area where it's possible the remains of a airplane that crashed in the early 1900s, the Samoan Clipper, was thought to go down. So we're double roboting tonight. We got two robots exploring two different parts of the seafloor in two different ways. Pretty exciting. And also, anyone who wants to jump in and describe what they're seeing, I know that folks who are watching online get pretty jazzed to hear whatever you want to talk about. I know I'm not supposed to peek over at your screen, but I do see a comment from Annie Hollick. Or is that's from, yeah, just for a couple minutes ago. I sailed with Annie about a year ago.
Hey, Annie, I did too. It's Dave.
She wanted to know how long this dive is and any specific objectives. So we have about 20 hours or so, or maybe 18 hours scheduled for this dive. And the objectives are to observe the benthic ecosystems, the habitats that are down on the seafloor, to look at the geologic features and maybe collect some samples of rocks and organisms for further study, and to utilize these the sensor and eDNA sampler that we have on to look at the CO2 concentration in the water and to sample for eDNA. And, as we are just getting started, all I can say is, let's go. That was Annie's tagline.
That was Annie's line for sure.
That's a cool looking squid in the Atalanta camera. Has anyone ever seen a vampire squid?
Not in real life, but I'm definitely hoping we'll encounter one during this expedition.
Only on the
It's
highlight
on my
reel.
bucket list.
Yeah, it's really cool. It has this like big hood-like thing that it opens and kind of shimmery and purpley black.
Okay. So someone on the chat wants to know about the quad cam. How's it going on the quad cam?
The quad cam.
Yeah.
So is this what is being fed on the quad cam right now, Jamie? Oh,
Not sure what we're talking about here.
what's going out on the quad?
We're not sending anything out on the quad.
Oh,
We just have three satellite feeds, Sat 1, Sat 2, and Sat 3. Sat 1 is the Hercules camera. Sat 2 is the Atalanta camera. And feed 3 is right now a picture of folks here in the van.
oh yeah.
And,
Is there not on the live stream a place where you can
there is not.
see all of them together? No.
No, there is not.
Oh, interesting.
There are other things that I can put up. When we launched, we have a new thing that we're experimenting with here, and I put
Ooh,
that
yeah.
up, so we could see the ROV going over and that kind of stuff. See the front row. Everybody in the front row wave.
Oh, yeah, they follow directions.
Camera's down there by Johan. So we've got that.
Is that called Partridge Family mode? That's kind of like how they did their intro.
It's just a multi-viewer. We have this same multi-viewer changes into a quad when we do sampling, and it shows the various sides of the ROV, vital box, manipulator, that kind of stuff. We're still experimenting with this stuff.
That's cool. That's it's, we're all kind of experimenting, right?
Yeah.
Like it's experimenting to put all these instruments on the vehicle. The scientists, of course, are doing experiments, but we're also experimenting with, how we transmit the visual information. That's the fun thing about being on this boat. You have an idea, you can try it.
Okay, now all you guys in the back row, look down towards Erica
Oh yeah.
and wave at the camera. Hi, there's the back row, the science row.
Where's the camera? I'm like missing where it
Right
actually.
next to Erica's monitor.
Oh my gosh, that's tiny. Cool.
So there you go. And then
Now, what about the one on the video station there?
oh yeah, it's broken.
Oh, it's broken.
Yeah. Now we've got. There you can see us in video. We're way down here, hiding in the corner, that kind of stuff. So these are new cameras that we just put in a few months ago.
They look really good.
Yeah, they do look pretty good. And then if you really want it, there's the video camera.
Ah, yeah.
Back of my head, best angle.
Okay, wave, turn and wave.
Hi, Mom. And there we are. That's good enough for that.
So we were just having fun back here looking at SAGE and comparing it to our oxygen concentration. So we were seeing a little increase in the carbon dioxide as we see the decrease in the oxygen as we're headed down.
Any ponderings as to why that is?
Oh, well, probably, because that's where all those mid-water critters have been hanging out all day, sucking up all the oxygen. And now they're up, and we're picking that up.
That's cool. So marine organisms, just like humans, take in oxygen and give off CO2.
And one of the cool things that we are looking at with this expedition includes the mid-water organisms, and that's what Annette and the multi-puffer are helping out with here is figuring out what is in that mid-water level. And it's part of the most amazing migration of animals every day, where these animals that are living down in the deep migrate up towards the surface at night so that they can feed and access all the phytoplankton and all the yumminess that it must be in the evening while they are free or hidden, not really free, but they are hidden from predators. So very cool that we can see that signature in the data as we go down.
Yeah, it's amazing. We get very excited, and rightfully so, when we see a big, charismatic marine organism like a shark or a whale. But the microorganisms are so much more abundant and really are the kind of heartbeat or life cycle that defines the ocean.
Absolutely. We've got algae, microbes like bacteria and other types of things living in the water, and those help feed all of the other critters that we see. So pretty amazing to think about how the ocean is really our lungs for the planet. And it's all of these small plants that are doing that on the surface. And then they feed so many other critters. As we were coming down, you might have noticed some small larvae that were in the water floating around. Those could be a number of things from crustaceans, fish, corals. A lot of animals in the ocean have a planktonic phase where they float around so that they can be spread out to new places. And so we got to see some of those on our way down, and now we're just waiting patiently to get to the bottom. We're at about 11, just shy of 1,100 meters at this point. Eagerly awaiting the bottom.
I don't know why, but I always like try and be the first person to see the bottom, like, oh, there it is.
It's like when you're on a ship for a long time and the first person to see that land over the horizon.
Yeah.
Although we're kind of cheating here because we have the depth measure counting us down. We know about where we're headed to.
Yeah, and when we get closer, we'll be able to tell how far we are from the bottom. We use I don't know exactly how we do the altitude here, but in general, the works on the principle of sending out a pulse of sound and seeing how long it takes to go down and bounce back up, and I can convert that based on the speed of sound in water to a distance. I think we use DVL for that, but there's also like certain devices that are just for measuring altitude.
I was going to say, do we have some, it looks like maybe a little jelly there on
Yeah.
Atalanta.
And if you're looking at the Atalanta feed, you'll see kind of this pulsing, which is the organisms are not moving that quickly. It's that we are moving downward, so it looks like stuff is coming at us, but we're also feeling the motion of the ship that's controlled by the swell or the waves on the ocean. So that ROV is kind of bobbing up and down. And the reason we have the two ROVs is so that one can isolate the main or the larger ROV from that motion of the waves. So that one can bob up and down while the other stays pretty steady.
It's kind of mesmerizing. Just kind of floats in and out, just like the waves.
So who on this watch has not participated in an ROV dive before? I guess who's going to be seeing the seafloor with their own eyes for the first time?
This is the first time I've been on the ship. I've watched the telepresence for years now, but first time I'm going to be on the ship waiting for the feed. What about you, Erica? Yeah? First time? Yep, got a nod there, yeah. Stefano, first time?
Yeah, first time.
Yeah. What do you most want to see?
I would say anything.
Anything.
Yeah, I guess
Yeah.
maybe what do you expect it to look like? I'm not even sure what it's going to look like here. Could be a lot of sediment, could be a lot of bare rock, could be a lot of animals, could be few animals.
That is the excitement of exploration, right? We don't know what is waiting for us down there. It's going to be, I'm literally like sitting here just waiting to see what we see.
Okay, I have a question coming in. It says, what sort of sensors and data are the pilots monitoring most during the descents?
You're muted, Robert.
So the big ones being depth and our heading, altitude.
How do we measure depth on this vehicle?
On this vehicle, we have a pressure sensor. So it uses a crystal oscillator, and the pressure on the crystal changes the frequency of the oscillator.
That's cool. Is that called piezoelectric?
Yeah.
I wonder what piezo means.
Has something to do with crystalline structure.
Yeah.
Bob, what's the difference between altitude?
Between what?
What's the difference between altitude and depth in your
So
water?
depth is from the surface to where going down, and altitude's from the bottom coming up.
There you go.
So do they use the same technology, the piezoelectric, to measure altitude when you're like flying in an airplane? Does it go, I know that increase in pressure will change it. What about a decrease in pressure?
Yeah, I think they set their barometer when they're at the
Oh
airport,
yeah,
right, to a
that's
known
right.
altitude.
Yeah,
Yeah.
that's cool.
Okay, you want,
It's also, so the two robots I mentioned, Hercules and Atalanta, are connected to the ship with a cable, and inside that cable are copper conductors for power and fiber optic cable for transmitting data. And so all that information that the sensors are producing, we get to see it in real time, including the video. And the other robot we're using is not connected to the ship by anything, so it goes off on its own, and this is the reason we can do both at the same time. So one stays with the ship, and the other is free to kind of go about its business. And so from that vehicle, we don't get nearly as much information in real time, or even in like slightly delayed time. We might send information acoustically through the water of how deep it is, and some small bits of information about what it's doing. But for the most part, it has to have some intelligence on the vehicle itself to avoid obstacles, to maintain its depth and its heading. So it's a great complementary pair of robots.
Yeah, Hercules and Atalanta aren't really robots, they're remotely operated vehicles. So as Sentry's an actual robot, it's autonomous and does its own thing.
Right. Is that, yeah. Well, I'm going to continue to think of them as robots incorrectly. Yeah, I guess it's like a vacuum cleaner and a Roomba.
Yeah.
Come on now, they're way too cool to compare to a Roomba and a vacuum cleaner. I mean, Hercules and Sentry, they are both amazing, amazing pieces of machinery. I'm very excited to have them out here in our waters in American Samoa this month.
Yeah, it's been cool to be in American Samoa, a place I've never been before, and just everyone was super friendly, and it's a beautiful place, and some things that I've never seen before, like these big fruit bats, and it's been really cool.
And we are approaching, we're at 1366 right now, still on our way down, so we're getting closer to the bottom. Hope everybody's patiently waiting to get there with us. Any bets on what we're gonna see first? I heard sand in the background.
Yeah, I'll go with rock.
Ooh, we got some sand, some rock. We're exciting stuff there. Any other thoughts?
An old airplane.
That'd be nice.
That would be really nice. We just magically drop down right on the Samoan Clipper wreckage.
I'm gonna go with we're gonna see a fish first.
Ooh.
See it
Oh, fish.
before we see the bottom. So.
Could go for
Yeah,
that.
we're gonna
Okay.
see dirt.
Okay, technically it's not dirt, it's sediment.
Is it mud? Is it mud?
Oh, we've got a jelly.
Sediment.
Sediment.
Yeah.
Or was it a jelly or a siphonophore? Didn't catch it.
Okay, as
I'm
the
moving too
geologist,
quick.
I'm counting on you to.
Sediment or rocks?
Uh-oh. Pressure's
We're gonna
on
get
now. I
rocks.
forgot about that. Oh.
You
Sean
see?
was saying there's lots of rocks.
Yeah,
Down
but
the rocks?
they're scattered. They're scattered through. This will be different because he's been on kind of flat stuff, and this is going to be sticking up a bit. But we'll see.
Oh, starting to get Doppler.
Oh, we're getting Doppler. We're at 14, 15. Getting closer.
So you get rid of the stick and do manual.
Okay, as we get closer, there's a question that says, what is the maximum depth of the AUV
So on your
Sentry?
altimeter,
Oh yeah, so Sentry's maximum depth is 6,000 meters.
you can stop at 30.
That would cover, that would allow it to go to, something like 90% of the ocean floor. There's not too much ocean that's deeper than 6,000 meters, and certainly most of the area around American Samoa. Past couple days it's been working at, couple thousand meters, but later on in this expedition we're going to be pushing up against its depth limit.
That's true. In the outer layers, outer areas of the EEZ, we get down to over 5,500 meters. So excited to have Sentry out here and be able to look at some of those places that no one has ever looked at before.
Okay, it's switching to DVL.
Yeah, it gets harder and harder to get to those places the deeper you go, largely because of the hydrostatic pressure just getting more and more.
I don't want to think about that too much. Kind of freaks me out thinking about all that water stacked up on you. Thousands of meters. As a scuba diver, we only go as deep as about 30 meters for most of our dives because of that pressure. I can't imagine
Do you mind if I do a reset?
what it would feel like.
And when you get down to 30 meters, do you feel it? Do you feel it like pressing in on you?
There we are.
When I'm free diving, we feel it definitely on kind of our lung capacity. You really feel that when you're free diving. Not so much when we're scuba diving because our lungs are filled with compressed air, so we have more to fill up that volume, but definitely when free diving. We're at 1486. Are you guys getting excited? Yeah?
Yep.
Yeah. It's coming. We're going to see it soon. Who's going to be first?
It's always the front row. They're closer to the screens, so... Oh, I see it.
Oh, there we go. There is the bottom, and there is sediment.
Sediments.
Lots of sediment. Props to Chris. He called it.
All right, so down on the bottom. That's very exciting.
Here we go. Welcome to the base of the Tutuila Ridge.
So we're going to take a few minutes, probably get set up down here before we take a look around, but get a good look at the seafloor, and this is pretty characteristic of the seafloor around here. This sediment you see is probably mostly what we call like hemipelagic mud, so all the material raining down from above, the marine snow collecting.
So we've gone from a blue screen to a, what is that, cream colored screen?
Ooh, I like that.
Kind of seeing some of that seafloor too from farther back with Atalanta. Kind of a wider picture view there. And we might have a critter there on the right. We just passed it.
If you're seeing the red button on our question chat box on the website, you probably just need to refresh or maybe clear your cookies, but the chat box is open, and even if you see red, please submit the question. We are open and want to hear your questions.
Where are these cookies?
135.
Hello, Annette here with the sampler. We're going to start sampling with pump 0.
You know we're on the bottom though. Do you mind sampling when we're sitting on the bottom? All right, go for it. So, Erica, they're going to start pump 0 on the multi-puffer. You give us a word when you started, Annette.
I've started, but it's not pumping.
Okay. So hold off on that.
Ready.
Wah, wah.
Trying again with pump 1. Pump 0, sorry. Trying again with pump 0.
I wonder what makes all of those kind of indentations that we're seeing on the in the sediment there from Atalanta. Any thoughts on that?
Yeah, I'm guessing organisms of some sort. You often see kind of wiggly trails through the sediment for the filter feeders. I don't know that I've really seen that kind of like footprinty pattern before, but usually any disruption in the sediment's surface is from organisms.
It does kind of look like big footprints or something, like kind of
Yeah, setting down there.
setting down across there. Interesting. I'm used to seeing snails up in the shallows. You know, I'm a coral reef ecologist by training and experience, so it's interesting to see the comparisons between places.
We're going to try with pump 2 on the eDNA sampler.
Roger.
Now.
Okay.
It's pumping.
Yay.
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 Hooray.
So we're in the process of collecting our first eDNA sample.
This is very exciting. These are going to be going to be the deepest samples so far for the expedition at 1,500 meters. Yeah, I'm getting a thumbs up over there. Excellent. Very exciting. So we're extremely excited about environmental DNA. Oh.
Sorry, can you repeat that? It's 02, pump 02.
Yeah, but in the numbering of samples being collected, it's the first one, 001.
Correct.
On pump 2.
Correct.
Roger that.
We're all getting sorted here. The first time we've reached the bottom on this this expedition, so bear with us as we're all getting settled in here and communicating, testing out some of the equipment and manipulator arms and such.
Hey, Johanna, when you have a chance, does that sonar go to like a shorter range?
This one here?
Yeah.
That's actually something I don't have control over.
No worries.
But you wanted to know if we can decrease the range here when we have time. Adam, was that a sample that I should mark down?
Yes.
Okay. Yeah, it's at 100. Yeah. So Adam, were you saying to make it a finer, like closer range?
Yeah,
Like
I
zoom
just
in more?
wasn't seeing it. I just
Yeah.
wasn't seeing anything, so I was wondering if... There we go. Oh.
Where are you? That's at 50 meters. Yep. 30. Great.
I'm fine. Mm. Okay. Mm-hmm. Roger. Ooh.
135. You're muted, Robert.
Hey, Dave, do you want the downlight on or off?
Show me what you got.
You don't care?
I don't care.
All right.
Maybe I don't care, but I don't know. What's on right now?
The down's on. You want it off? There's off.
I think off for now,
Off.
until we're working close up or something. And I did a white balance of sorts. So we're good to go.
Stupid auto altitude.
Okay. So RV, you guys are set?
Yep.
Great. Adam, should we get going?
Yeah, let's head towards, is it waypoint 2 that's the next one?
Yeah.
Yeah.
All right.
Yeah.
I'll start with just a 30-meter step with the ship.
Yeah.
All right. Bridge, bridge, nav.
What's that on the seafloor there, Bob?
Hi, can we move the ship 30 meters at 135,
You
please?
zoom in, Dave?
Copy, 135. Thank you.
Oh. It's the edge of a rock. Or no, or maybe an old sponge stock.
Yeah, that's what it looks like.
Oh yeah, do Nautiluses live here? Yeah. Do they live around here? Oh, that's cool. Just hearing from Santiago Herrera that that Nautiluses live around here. We could see some shells on the seafloor. I'm good with that, Bob. Whenever you
Yeah,
wanna.
I'm moving on.
All right.
We're kind of out in front already.
Okay. Yeah. You noticed that as soon as there was a ship move, I was like, hey, let's stop and look at something. Dave, can you remind me, this the main camera doesn't tilt, does it?
The main camera.
The main...
The Herc camera?
Yeah.
Yeah, it does.
Oh, it does?
Yeah.
Oh,
That's
could you...
That's Bob's control.
I see. So you can kind of see a little bit of ripples in the sediment. That means that current's been moving across and piling up the sediment in little hills, and those, if we watch those ripples for a really long time, we'd probably see them migrate across the seafloor.
Robert, I'm going to give you another DVL reset.
Boop. So as we traverse up this slope, we'll sometimes see steep parts of it. We up to 30, 40, 50 degrees, and then probably some flat parts. Usually these are, even if they look smooth from the bathymetric mapping, there'll be kind of steps along the way. Those steps will have lots of current flowing along them, and that's where we'd expect to see high concentrations of these organisms that attach themselves to the rocks. Are generally all filter feeders, and so wherever the currents are, they're particularly happy.
Bridge, nav, another 30 meters at 135, please. Thank you.
So all that kind of dark specks within the sediment, I expect is kind of organic material that's fallen down. The first thing we looked at was looked like a stalk of a sponge. Really interested to see what fish we might see. I don't know what the kind of endemic fishes are here in this part of the world. Oftentimes in the deep ocean, we'll see these eel-like fish, cusk eels and the like, that are, particularly adapted to low light conditions and kind of sparse food supply. And of course the cold temperature and the pressure. Temperature outside the vehicle here is, let's see, where is that on my monitor? I can't find it. Anybody in the front row know what the water temperature is?
We were looking at it earlier. Maybe once. We'll look at it again in a sec, maybe.
Okay.
It's
Will you pull up the
2.70
water temperature sensor?
degrees Celsius.
Just take a peek.
Oh, yeah. 2.7 degrees C. That's pretty cold.
Yep, that's what we're getting as well.
Awesome.
Yeah. You're muted again, Robert.
I think he said something really clever.
I was just saying that the temperature probe, the oxygen sensor, and the CTD all agree, like within 0.01.
Oh, that's good.
Yeah.
Is the temperature probe kind of calibrated for a certain range of temperatures? Like, what's the highest it could measure?
The max on it?
Yeah.
It goes over 400, I think 450.
Wow.
Yeah. So that's pretty good.
That's pretty good. I don't know that we've ever found anything hotter in the ocean.
So they were actually burning up the temp
nav,
probes.
another 30 meters at 135, please.
What do you mean?
On the ONC cruise, they burned up some temp probes.
Thank you.
Oh, really?
Yeah, because it turned out the ones that the sensors they were buying weren't the 450 degree variety.
Oh my gosh. So did they just peg, or did they like actually melt?
They ruined them, destroyed them.
Was that earlier in the season?
Yeah, at ONC.
Yeah, yeah, I remember that.
They were scrambling to find temperature sensors.
Yeah, salvaging anything we could find.
Yeah.
So we got a bunch of different ways to collect samples on this vehicle. We can use the manipulator to pick a sample up and put it in one of the sample boxes that are arrayed around the vehicle. We can close these tubes called Niskin bottles and trap some water in there. We can suction up a sample with a suction device that will put it into a little canister. I think that's probably, and then we can filter for environmental DNA. On feed 2, you can see the view from Argus, which is up above Hercules, and kind of get a sense for what's around the vehicle. Right now it looks like not much, but as we approach the slope, I'm sure we'll be able to see kind of outcroppings of rock around the vehicle.
Which? Oh, you should do it once an hour. But in general, you should pay attention to the gauges. Like, yeah. So that's, oh, so you gotta, here. You got your camera control here. Oops, dang it.
Bridge, bridge
bring
nav,
that up.
another 30 meters at 135, please.
This
Thank
thing.
you.
Oh, that right there. And then you can zoom here. But I think, like, that's, yeah. Well, no. So gauges, and then you just gotta zoom in. The other zoom in. Yeah. Anyway, you get the idea. And then you can, yeah, and you can move it if you go in the middle of that box. There you go. You're all over it.
Yeah, someone on the scientist short chat asked us about the round holes in the sediment. I did notice we passed over something that looked like kind of just little collapsed holes, but haven't seen them since then. We'll keep an eye out. If we find one, we'll take a look. For all the
We're 3 meters off the bottom.
That's good. For all the CO2 heads out there wondering what our CO2 concentration is, you'll be happy to know we're holding steady at 280 microatmospheres. Did I just kind of go into disc jockey mode there for a little bit? Like, a little bit? Sorry.
I was still thinking about CO2 heads, man. Wow.
But the funny thing about the CO2 is, like, it's pretty remarkable. 5 years ago, you couldn't know in real time what the CO2 concentration is, so very exciting. I'm super excited to use this get over to Wailu'u, the active end of the Samoan hotspot chain, where in the past those fluids coming out of the seafloor have been saturated with CO2, even to the extent of producing bubbles of CO2.
Bridge nav, another 30 meters at 135, please. Copy, thank you.
Can we zoom in on that shrimp?
Shrimp? Where is it?
Kind of in the middle of the screen.
You zoom in, Dave?
Oh. Okay. That's cool. And what is that other gooey thing? We don't have enough biologists on this watch. Look at that thing go. Wow.
Whoop. There it goes.
All right, looks like I got back from learning all about the eDNA samplers just in time to miss the shrimp.
I think there'll be
Looked
more.
like I was a pretty good swimmer, huh?
Well, yeah, directionally, I don't think it was a little challenged, but but it can move.
I definitely did.
Yeah,
Saw it out of the corner of my eye, and next thing I know, it was gone.
Right on time, Val. There's a question from Norway. It says, can you explain what DNA is? eDNA, please.
Yeah, absolutely. So eDNA is environmental DNA. And so what we mean by that is that there are little pieces of DNA that are floating around in the water. And this could be from skin cells, it could be from feces, it could be from partially digested materials that are floating down. And so any type of DNA that is shed. So I think everyone's familiar with swabs that are used for DNA. And this is just DNA that's out loose in the environment. And so what the eDNA sampler is doing is collecting those little bits of eDNA, and then they'll go back to a lab where they're going to be matched up to known sequences of DNA. So I think if folks are familiar with the concept of DNA, it's kind of the biological code. It's the code that makes up all of living creatures, and it is made out of specific molecules, and they follow a pattern. And so each species has some parts of their DNA that are unique to them. And scientists have been collecting those over the past couple of decades and now have these big libraries. And so by collecting this DNA out of the water, we can actually match it up to known creatures and be able then to tell us what is here. And so even though we might not be seeing anything right now, we're currently running this multi-puffer to collect eDNA, and we'll be able to see what is down here, maybe off range of our cameras or something that has passed through just before the ROV came. And that's going to be really exciting because this eDNA is going to give us a real glimpse into the biodiversity around American Samoa. And there's been very limited eDNA samples so far to date taken from American Samoa waters. And so this is going to be a huge advance for us to really kind of figure out what is in our waters, and that we're not necessarily able to see with ROVs. So very exciting, and thanks for the question.
Thank you, Val. Appreciate it.
Yeah, there's another one of those little collapse holes. Oh, and an organism in there.
Yeah, look like we might have had a crustacean there to the right. Oh, maybe an anemone in the hole. Part of me wants to stop and look at every little thing, but the rest of me wants to move forward and see if we find something more exciting.
Oh, we got
Is
a
that
fish
a fish
coming
up ahead?
Yeah.
Ooh. Oh, here we go. And of course, at this moment, I am blanking on my deep sea fish identification.
Wow.
You bring me along for just
zoom
this
in, Dave?
reason. I'm spacing it.
Let's see. It could be.
Looks like an eel.
Yeah, there's a lot of eel-like fishes.
There certainly are down here. Until we get you.
Probably in the macroeuridae family, maybe. Oh, where's Megan when I need her?
Right, I thought I'd studied. I haven't studied enough.
So most of the fishes down here are kind of scavengers, yeah?
Mm-hmm.
That's a beautiful one, though.
Look at that imagery. Camera's really nice. Oh, it just picked up a little something there.
It's going to throw it at us. If
And
we
you'll
get
notice
closer.
they're
Oh
not.
my goodness.
Ooh.
Oh.
You notice it's not as colorful as our coral reef fish here. That's because there's not any light down here. So they can spend their energy on other things rather than looking pretty.
That's my philosophy.
Bridge nav, another
Ooh, there's
30
one of those
meters at 135.
holes, or a series of holes. Some organism is kind of a burrowing organism down here.
Copy, thank you.
So that what I think of when I see these kind of burrows is some scientists like to sample sediments from the seafloor to kind of understand what the past history of climate on the planet has been. And one of the things that makes that difficult is bioturbation. So there's a lot of animals working their way through the sediment, kind of mixing it up so that an individual layer kind of can move vertically through the sediment as the animals will kind of ingest the sediment and excrete it in a different place. I think we've seen a number of leafs think there's leaves on the seafloor here. And there's cool rings.
Couple of rings.
Yeah, you saw those?
Mm-hmm.
Oh, there's a little holothurian or sea cucumber.
Zoom in
Nice.
on
I
that,
love
Dave?
the deep sea cucumbers. Let's see if we can get a little closer look. Some of them are so pretty with their pink, translucent kind of skin.
Yeah, you can often see their digestive tract, and they're just scooping up that sediment and sucking all the goodness out of it.
This would be a good vacuum cleaner analogy
Oh,
here.
yeah.
This is exactly what these sea cucumbers do. They're feeding on all of the detritus there in the sediment, and they digest that, and basically clean sand comes out the other end.
Mm-hmm.
They're literally like the vacuum cleaners of the sea. Very important activity that they do there for us. Important service.
So, Robert, is the seafloor sloping up a little bit, or has it been pretty flat?
I've been in auto altitude, so I don't know.
Hard to tell.
It's
So that
supposed
is
to.
a robotic function there.
There you go.
It should start ramping up as we move along, but it's still pretty flat for the next couple 100 meters.
Okay. And Val, are the eDNA people, have they left you to?
Bridge nav.
They
Another
have, so I'm
30
going to be doing
meters
double
at
duty. I'm going to
135,
be running
please.
back and forth to our eDNA sampler and checking it. So we're
Thank you.
collecting samples the whole dive, what's called a gradient. So we'll just keep collecting as we go. And then if we find anything really interesting, we can take a sample at those particular places, either with the sampler, which is passing through a lot more water and able to give us a much more detailed sample, or we can pause with the Niskin bottles and take a more discrete sample.
And are we currently pumping for the same original sample, or have we stopped that
Yes,
first
we are
one?
currently still on our first sample, and we will pause that once it gets to the desired amount.
Okay.
I really like the top-down views of Hercules we get to see on our screen from up above
From
with Atalanta. Yeah,
yeah.
it's always fun to see Hercules down there doing its thing.
There's another one of them fish.
Yeah, so our initial thought on this is that it's a halosaur, so it's an eel-leg type of fish, so it's a bony fish related to a lot of the fish we're familiar with up in the shallows. As opposed to a cartilaginous fish, which would be like a shark or a ray. So,
You want to zoom in on him?
yeah, if we could zoom in and get a
Zoom in,
little view
Can
of
do.
that,
It's
see if
Samantha on
it's the
cameras
same.
now.
Maybe it's same species, or maybe it's a little different.
Getting in a little tighter.
Looks pretty similar.
I think the previous one had more of a dark head.
Do you want to get a picture?
I don't know if the coloring matters so much. And it had less opaque side fins. Pectoral, is that what those are called?
Yes. I like how sinuous that tail is on this, this fish. Do we want to snap a picture with our fancy camera here?
Yeah,
Got the new cameras going. We just take advantage.
You want to use the WCA cam?
Yeah, I'm looking at that.
You need more light on the subject here.
Can you tell if I'm focusing in the right direction? Let me autofocus that. All right, bam.
Let's see if we can get one
Bam.
with it. Is it possible to shift so we can get the tail in there as well as that?
the camera we're taking the still photo with is
Okay, the one down there.
Yeah,
Got it.
Are you
Excellent.
controlling it?
Yeah. Yeah, look, I'm zooming in. Oops.
It's a good thing Adam's on the controls, not me.
I gotta focus, hold on.
I'll pull out a little bit so you can recenter.
No, I have control of this one, unfortunately.
It's always a learning curve with new tools that we're using as scientists. So.
Oh, frick, that's such a good setup. Sorry, Bob, I'm trying to get focused here.
We're about 15 meters behind Anna.
Okay.
Are you struggling to make it focus? Or what's
yeah,
up
exactly, yeah. So.
Operator error?
No, it's a stupid.
It must be the camera, must be.
I'm not seeing any change in the focus as I turn this knob. All right, bail out, bail out.
All right, I think I gotta get going here.
Yeah, go ahead, go ahead. I'm not sure why it's not focusing.
Snap a picture.
Oh, never mind, I got it. Push is autofocus. God dang it.
Bridge nav, another 30 meters at 135, please. Thank you, Flavio.
Got it.
And I see we have our laser pointers on again. Did you guys already talk about that?
Nope.
So those are going to help us tell how big things on the bottom are.
Well,
and
not right now because they're grounded.
But they're on though.
They were on?
Yeah,
no, they're
I was
off.
just checking them because they were grounded earlier. And then
Oh, let's slow down for this urchin here.
Yeah.
He's teasing us with the lasers and then taking them back away.
Zooming in over here.
Ooh.
Oh, interesting.
That might actually be, is that an urchin or is it?
It's an urchin.
More of a.
It has the radial symmetry of an urchin.
Oh, there we go. Yeah.
Look at that.
So let's see. So part of what we do on these expeditions is collect samples for folks who are on shore, and trying to look if anyone has any urchin requests. This is an invertebrate, yeah?
It is an invertebrate. It's an echinoderm, and
so you want to go for a first sample here, Bob? Or is your manip out?
Manipulator's not working.
Yeah, but okay. Let's hold off on that. I could see doing a little suction transfer, but maybe not right out of the gate. It's gorgeous.
Yeah, look how fine those spines are on it. And it's also moving. You can see if you look real closely, they also have some, you can see it can manipulate some of those. Might be some little feet on the bottom as well as those spines. So you can see down there, there's slowly